Yeah, my name is Jimmy Payyappilly. I am from ECCB BI in Cambridge, and I'll be today walking through some of the Global Alliance for Genomics and Health, also known as GA4GH standards, which are interoperable, and how we can use them in genomic data sharing. I would like to also take the opportunity to thank the organizers for this chance to share this. So today I would be focusing on four major standards that were developed by GA4GH. One is Data Repository Service, the Workflow Execution Service, the Task Execution Service, and Data Connect. Each of these standard actually helps researchers and users in specific functions of their day-to-day tasks. It could be data storage, it could be modeling, it could be searching, or it could be pipelines. So these standards help build an ecosystem which is interoperable and can be used in your infrastructure quite easily. So we have heard about a lot of today there was a lot of talk about tools, about data. What we are trying to do here is to standardize the data sharing such that it can go across ecosystems, go across infrastructures without needing too much of investment in getting something up and started. That's the ideal situation, and this diagram presents exactly that. What we envision in GA4GH, the ideal ecosystem would be that there is a CAD controlled access data source, such as EGA, for example, having data on the local system or on the cloud like S3, which is accessed by an indexer function, which uses appropriate access permissions such as tokens, and it takes up the metadata from the controlled access data CAD and converts it into a readable format, CSV, for example. This CSV would have information on such as the IDs and different information of metadata about the files. And that data then is served into a DRS database instance. At the same time, the same data is also cataloged into a data connect instance. Now, when you see the global GA4GH emblem, it means these are the standards that are being used. So the data connect instance would help in cataloging the data which has been retrieved from these CAD sources. In the manual part of the execution, when the researcher triggers the workflow, WES and TESS, again, two standards, will handle the flow, will handle the execution. They would look up for data by triggering a hook into Data Connect instance. From there, Data Connect would then supply them the data it had initially cataloged, which is a list of IDs, which then the test WES and test instance can connect to the DRS instance, and get the file path information from which the further computation analysis by the researcher can go on and then finally return the results to the researcher. So this is the ideal framework. This is what we envision would be where everything works end to end, seamlessly, interoperably, where you don't have to worry about data massaging, you don't have to worry about different formats. It's all connected together seamlessly. Now let's come back to the ground, back to earth, real life. At Humble ABI, we are trying to do the same, and we have gone well, I would say, from the implementation, which I just showed you. First, we are using the 1000 genomes open source data, also known as IGSR, as the CAD source. The same indexer function retrieves the metadata from these files and it converts it into a CSV. This is an actual snapshot of the CSV that's generated. You can see it has ID, it has the file path that's pointing to the CAD source, the types, the size, and different parameters like checksum of the file. This is what is passed into the DERS starter kit, which is a lightweight implementation which helps to store the data into the DRS instance. Now, from the manual perspective, the researcher, in this case, for EBI in the network, triggers a CNS script, which then triggers the workflows, next flow workflows, which are hosted on the Slurm. The Slurm then gets a list of the DRS IDs, and it triggers this back to the DERS database to retrieve the file paths, return it to CNest, and send the results after computation analysis, sends the results back to the researcher. As you can see, in a year's worth of effort with just one developer working on this, we have been able to deploy easily within the EBI network. Not on prod yet, we are just getting there, but a DERS instance, that is an interoperable instance. The next step for us would be to convert this further and go down the path of ensuring the other components such as REST, test, and Data Connect. Data Connect would also be integrated. So quickly getting through my last slide, promise. So if all of this sounded interesting, yeah, there are quite few people in the crowd who are actually known to be part of GA4GH. So please, if you would like to contribute, these are the GitHub links. I'm available downstairs on poster 129. Please come have a chat and we'll be happy to help you get started. Thank you.